Okay, the last part for the bacterial growth. Okay, remember the bacterial growth, smaller in size on my whiteboard. They are experiencing the binary fissions. So this is basically the situation for the bacteria to grow. Okay, so once the bacteria wants to grow, it will start with the lag phase. So what do we mean by lag phase? It needs to acclimatize with the medium that they are in. Let's say they are in water, so they would like to acclimatize with that medium. You get used to it, just like you are entering UTM. Okay, you don't simply get so excellent. Okay, you want to acclimatize with the system. Oh, this is the way that people study during the undergraduate time. So you need time. So that is the period. Okay, we call it as a lag phase. But once they have adapted with the situation and the foods are abundant, okay, there are so many organic compounds and inorganic compounds, so they can eat very, very happily. So they start to grow exponentially. Okay? That's why you can see a very big jump suddenly in growth of bacterial populations because of the food abundance, and they just get so suitable to reproduce themselves. But at a certain stage, whereby at this particular time, we call it as a stationary phase, okay, the food is no longer abundant. And they observe that the number of cells die is equal to the number of cells grow. So they cancel each other. Dying and also growth cancel each other, and that's why they have a stationary phase. So no increase in growth or no increase in the bacterial population. But once the food is very small in amount, or basically the rate of death, okay, exceed the production of new cells, so you will be observing a death phase for the bacterial population. You can see the amount of the bacteria survive is getting lower and lower as the time goes by, as the death has exceeded the growth of the population. Okay, so this is basically the bacterial growth curve that you need to remember and to understand. So I think the explanation that I already mentioned is being written here. So that's basically that you need to know about the presence of microorganisms in water. So what are bacteria? What is the size? What about viruses? Okay, what about algae? What's the difference between algae and fungi? What about protozoa? Is it a smaller microorganism, or is it bigger than bacteria? So those kind of things. And what about rotifer? What does it mean to have rotifer in your sewage treatment plant? Is it good or is it bad? And the last part is you are learning on the bacterial growth curve. Okay, so they have 4 phases. The first one is lag phase. This is when the bacteria get acclimatized with the surroundings. And then exponential growth phase, whereby they have abundance of food or abundance of substrates for them to enjoy and start producing very, very high. And once they reach the stationary phase, whereby the new cells growth is equal to the number of cells die. Okay, that is what we call a stationary phase. So you don't see the increase in number of microorganisms, or you don't see the increase of number of the bacterial population, and eventually they are forming or they are reaching the death phase, whereby the number of population of bacteria is decreasing as the time goes by. So that's all basically.